Hello, everyone, and welcome to today's Keynote webinar titled Pathogen Discovery and Diagnostic Tools for Pandemic Preparedness and Prevention. This webinar is part of the 9th annual event in the Microbiology Week virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I'd like to remind everyone that this event is interactive. So we encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits image located on the bottom right of your screen and follow the process to obtain your credits. All right, now let's get started. I'd like to now present today's speaker, Nischay Mishra, Assistant Professor, Epidemiology, Center for Infection and Immunity at Columbia University. For a complete biography on our speaker, please find it on the left of your screen. Nischay, you may now begin your presentation.
Thank you, Kelly. Melanie, who has been helping me to setting up with this fantastic web page. And thank you, Lynn, for inviting me, and LabRoots. I'm happy to be here. I hope you are hearing me clearly. I'm Nischay Mishra. By training and by experience, I am virologist and infectious disease scientist. I have been working on outbreak and pandemics and epidemics for a long time. I'm trained virologist. Right now I'm working as assistant professor at Columbia University Center for Infection and Immunity. So today's topic, I will be talking about tools. Those are important for the discovery and diagnostics for microbial infections and diseases, and their capacity and their expansion for the outbreak prevention. Why do I say outbreak prevention and containment? Because every pandemic starts with a smaller outbreak. So if we can identify the outbreak or smaller epidemic label, we should be able to prevent the future pandemic as a first tool. Like earlier, the prevention, better the preparedness. Thank you. Okay, so what we do, I work at Center for Infection and Immunity. We work on all kind of microbial diseases, viruses, bacteria, fungi, tick-borne diseases, any new pathogen discovery. We work in the wildlife, we work in the human sample, we rediscover the viruses and bacteria, we identify new pathogen as well, and we do characterization of them, develop diagnostics, take them to the approval from the agencies for regulatory purposes, clinical diagnostics, and we also work on serological devices, and we also see roll up like microbial infection, chronic diseases like autism, ME, CFS, long COVID, dementia, Alzheimer's, diabetes, etc. And we have been training and capacity building for a long time, more than 25 years. But last couple of years, we have given this a name as a program that called GAP. I will talk about that as well. So as far as we remember, like last 10 years, like if we talk about it, there's several outbreaks. SARS-1, 2003, it's been long time, 20 years. But if you consider there is a like constantly every 1, 2, 3, 5 years, there is the emergence of a respiratory virus, bloodborne pathogen, some superbug of bacteria, some new fungal that is very infectious. It's just keep happening. There's several reasons behind it. I will not go into those details, but as we know, shifting overlap like niche of from human to wildlife, deforestation, change in climate, more interaction, sometimes regulatory slippage, things happen, and then this higher population, more travel, one of the most important point of view also. When people travel, they can carry pathogen with them, and then or they're more wildlife or more poultry and all of those products, they've been like shipped around the world, so there's increased the chances of like human and wildlife interaction, zoonotic diseases, more infection, more mutation. Virus and bacteria can adapt quickly from different organisms, different pathogens, so that could happen. What does it happen like? Sometime, like if you remember, uh, when the SARS-CoV-1 happened in 2003, it took like 6 months, 7 months to identify the complete genome by several lab work together, because there was that time we did not have tools. But in recently we have discovered and developed like tools around the world, high throughput sequencing tool, high throughput serology tool. Things are easier to identify new pathogen. We are very good at computational uh, analysis as well, and bioinformatic analysis. There are tools that can tell us, oh, this virus look like that virus. It has a receptor that can infect X, Y, and Z species. So, so work has been more, but like so once we have like more tools, we have been discovering multiple viruses, and all propose different kind of risk. Some are like global, some are like very centric to one or two countries, some are like seasonal, and some are like reoccurring, like Ebola and Marburg. It happens once in a while. And then another problem is endemic viruses and bacteria, which always circulate with us: respiratory virus, gastrointestinal viruses, hepatitis viruses, other bacteria with common pathogen. They can occur during any scenario, and also antimicrobial resistance. So these things are very cumbersome. Some of those viruses, they can be worked up in the normal biosafety level 2 lab. Some can't. Some has to be handled in BSL 3. Some has to be BSL 4 lab. And the older technology, what or the older tools we used to identify the virus, were very prolonged like process. We used to culture the virus, look for the CPE, look at different electron microscopy, and then we kind of identify virus. But it's very difficult to distinguish if we identify the new virus in the electron microscopy. It is the same SARS-CoV-1 or SARS-CoV-2 until unless we have the data for both, or it's some new coronavirus, and we don't know if it can infect the human population or it can jump around from different species. So getting the genomic information is most important. So that contrast we have seen in SARS-CoV-2 time, when the first earlier case were identified sometime in mid-November, December, and when they worked up on the genome, within 2 weeks of that start point, around 7th of January, we had full genome of SARS-CoV-2. What does it provide? It provides first, it's good to know your enemy. It tells you the genetic information of the virus itself. We know how far it is from the previously circulating viruses. Also, it helps like providing the tools for the diagnostics and a early kick on a start to work on the therapeutic and vaccine and other models. So if we had these kind of tools, or we have this kind of approach, then we could change the scenario. We could change the path of many other zoonotic or emerging diseases in the past, like HIV. It discovered like around 10 to 15 years after, when it was circulating, and other viruses as well. So in this presentation, I would like to talk about how we can approach when the new emergence of any pathogen is happening, viruses or bacteria. I will focus only two today, and how we can build a system that can help throughout the world, and what other tools we have. I have developed at the Center for Infection Immunity in last 10, 12 years. So we're going to talk about that. So that what I was saying. If you see, there was an older scenario. There were influenza viruses, pox virus, West Nile viruses, hepatitis C, dengue, MERS, chikungunya. All of these viruses has been identified one part of the world or another. But if you know now, recently chikungunya has been spreading around not only in Africa and Southeast Asia, now it's in Southern America is also, and there are chances it could have circulating in south part of United States as well. Dengue, Zika, all are like surging through. There was a time when dengue genotype 1 to 4, they're different genotypes, they were restricted by the geography, but now all four genotypes can be found in all part of the world. So first virus emerged, then spread, and then make a settlement on that. Some are vector-borne viruses which need a vehicle to go from one person to another. Those are arboviruses. They mostly in hotter and humid climate because mosquitoes need that kind of atmosphere to survive. Some are respiratory viruses like SARS, SARS-2, influenza. They can travel from one to another without any vehicle. So we have seen several viruses in the past, not only uh, health public health point of view, fatal loss in the life loss. We have seen the very big economic loss also that also lead to other repercussions also. And then after infection happens, there are aftermath as well. Some people lead to the chronic post-infection disease also. We're gonna if we talk about that as well. But in last 3 years, the only we talk about the emerging viruses, SARS-CoV-2, because it in 120 years of lifetime, it was unprecedented how the virus spread, how many life has taken, how many efforts has been put to curb down that virus. And virus is mutating, is still making the new variant. But since we everybody have been vaccinated, immunized, or have been exposed to viruses, the SARS-CoV-2 infectivity is still very high, but we do not see high mortality. We do not see the high rate of severe COVID-19 cases anymore, which is a good news. And then there are some rare zoonotic viruses. The spread was contained timely. It could happen, and like it was identified, happened in the last 10 years. It will happen very quickly. Within 4 or 5 people of in family or a small village, they have been infected by the virus, and then it's identified, and then the whole chain of transmission has been broken, and then after virus was shut down over there. Spondivani virus, Sinombre virus, Luzo virus was identified in our lab as well, where the mortality rate was very high, or it was very infectious virus, but it did not spread beyond that small geographical area or small containment zone. And then there are like regular viruses, how they affect or how they infect the healthy individual, and how they're gonna infect people who are living with immunocompromised, immunosuppressed, or like people with solid organ transplant, cancer patient, etc. The pathogenicity is totally different in those individuals. Um, and 5, 6, 7 years ago, people used to think there are like 10 to 15% people in the world they have like immunosuppressed, immunocompromised status, but the number could be higher than that because not everybody goes to a clinic, and it's very difficult parameter to identify who is immunosuppressed, who is immunocompromised. People can have some sublying conditions, they can have some comorbidities, may have diabetes which has not been identified yet, or they have high cholesterol, and these all things make virus more in survival phase. Uh, are my slides are visual?
Yes, we can see them.
Okay, I see the question here. Slides are not progressing. Okay. All right. So the 3 point of like how we identify the new viruses, how we discover them, and once we discover them, how we deal with it. The most possible method is qPCR method, real-time PCR, multiplex qPCR assays. All of these assays are like feasible only once we have identified the virus and characterized the virus. So in last 5, 6 years, I have worked on several outbreak, but three I will be notifying. One was a Ebola virus outbreak, Zaire Ebola in 2015 in the Africa, and and then after that Zika virus outbreak in Southern America in 2017-18, and SARS-CoV-2 recently. So all three multiplex diagnostic panel we developed, we got them through the regulatory approval and for the diagnostic clinical purpose. Uh, how do we lead to that? It's all possible when we are dealing with the some organism which has already been identified. We know about it. We know the genomic sequence. We know the conserved domain for that. So we can develop these diagnostic assays. So in West Africa panel, we have Ebola, dengue, Plasmodium, West Nile, typhi, Shigella, yellow fever, and some housekeeping gene. Zika, we have Zika, West Nile, chicken, dengue, and chikungunya virus with a housekeeping gene. SARS-CoV-2 is a very distinguished virus, so we had only SARS-CoV-2 only, and it was emerging like the highest number of cases. So we did not worry about SARS-CoV-1 because which has been almost we have not identified any cases for SARS-CoV-1 in the last 20 years. Second, I will talk about two high throughput sequencing method which we developed at CII. They are direct detection of molecular target of viruses and bacteria, microbials. One is vircapsic that's for virus detection, and second is vircapsic for bacterial detection and antimicrobial resistant gene detection through high throughput sequencing. And third one is once like the acute cases are over, we would like to see through the antibody, which is very important tool in zoonotic disease. We want to see if this virus is circulating in some bats or some rodents in one possible geography. We do want to know if these viruses have been exposing the people living around it or handling these animals and have been exposed, did they produce an antibody response? Were they immune or not, or they're getting sick about it? So it's very important to look for historical infection, looking for the subacute infection, looking for the immune response. We have made a very high throughput serodiagnostic tool that identify the key epitope regions in the whole viral or bacterial proteome that can use for the recovery, past infection, without having any cross-reactivity. So first I will talk about real-time PCR diagnostic qPCR assays. This is the SARS-CoV-2 assay. Once the virus starts circulating and we started knowing the reports, there's a new coronavirus in early January. We identified the genome of that virus. We aligned it through the other coronaviruses. And in February, I had this multiplex diagnostic assay because how it was surging through, we were not sure how virus will mutate. And also we knew the number is going to increase, so we needed some multiplex diagnostic assay so we can take the full potential of any diagnostic assay where we can test for 2 or 3 targets in the same well, so we do not occupy the whole 96 well plate, and we can increase the fold for 2 to 3 fold. And we also needed a housekeeping gene also, because in the cases of outbreak or epidemics where samples are being collected throughout the multiple position locations, sometimes the cold chain is not maintained or they do not have facility in many places where samples are collecting. Swab is collected in the VTM and left around for a couple of hours. So we would like to know when we do the qPCR if the sample has been properly maintained and the nucleic acid was still intact. So having a housekeeping gene that is part of host genome RNA also very important. So we also added RNA-P, which is a human RNA-P, which is a housekeeping gene. So all 3 targets can be tested in the same well at the same time with the highest sensitivity we could achieve. We have made the primers and probe along with in the method. When all the primer and probe combination, they can work with each other. Here we see only 2 targets, but actually when we look at the biology and the chemistry of our triplex assay, there are several 5 or 6 different reactions are happening at the same time. The forward one can interact with the reverse one, and forward 2 can interact with the reverse 2. So there multiple target can go around on that. And also then we got the pre-approval in sometime in March, but final approval for emergency user authorization in May 2020. We had a very good limit of detection. We have positive percent agreement. We have very good 100% negative percent agreement. Like I said, when we started it, there was only one genome was available, which was published by China CDC in January, first week, 7th of January. After that, as all we know, the virus have emerged. There are several million genomic sequences still available. My target was having the nucleocapsid gene because which is mostly conserved domain of SARS-CoV-2 genomic structure. There are not as many mutations like we see in the spike gene. And so far, our assay work efficiently, very well with all emerging variant and all the SARS-CoV-2 variant which have emerged through in last 3 years. When we started this assay for any clinical setting, a lab working 24 hours cannot handle more than 1,000 samples per day because of real-time PCR extraction and manual setting up the qPCR on. We need multiple platform and everything. So it was a capacity constraint, and as all we know, there was shortage of qPCR, and many of like organization, they were overcharging for simple qPCR somebody needed in urgency, and the delay was 4 to 5 days, which is not very effective when we want to contain the any major outbreak or such kind of pandemic. So I collaborated with a company called Summer Bio. They have been working on technology and tools. So we worked with them. We made this whole system automated, all the uh, liquid arm and machine arm based system, which can extract more than 200,000 samples a day and do the qPCR. They're all the chain reactions. If only the same master mix, you make it in the liters, not in like small microliter tube, and they could test like around 28 million samples in so 26 months of when they were processing the sample in part of the California, LA, and then Southern California and some other vendors throughout the school district, and they were providing like 200 up to 200,000 samples per unit within 24 hours turnaround. Uh, in September 2022, we have shut down the facility, but still we have machine, we still have processor, we still have the technology. In the future, if something re-emerges, we should be able to develop the qPCR quickly and transfer it to high throughput sequencing method, high throughput testing method. In 2017-18, I also published this paper, and then I also got FDA emergency user authorization for Zika virus that can test Zika, dengue, chikungunya, West Nile virus all together. These are all arboviruses, present very similar clinical symptoms on the onset of that. So it's important to have something that can test them all together. Mostly of these diseases spread in the countries where they have a smaller structure, or they cannot afford to test 4 targets for the each sample. So that was the whole point developing a qPCR assay that can target all 4 viruses. West Nile virus is included because there were chances the Zika can spread to the part of the United States as well. And we were testing we were using this assay to test the samples from New York City Department of Health, State Department of Health, and NIH. When we work on like viruses like which are from the same family, like West Nile virus, dengue virus, and Zika virus, they come from the same family. They can show the very major cross reactivity. So it's important to test multiple targets at the same time and look at the interaction between different targets, because we do not want one primer to bind with multiple viral targets if they are present, and then the interaction uh, lead to the lower sensitivity or specificity. So we always use multiple target and we look at those combination. Ultimately, I ended up using 3' UTR for Zika virus and 3' UTR for dengue virus. West Nile virus, I use NS5, and chikungunya, which is coming from different family, we use NSP2, and housekeeping gene, it is HMBS, which is part of hydrom without benzoxine, which is part of blood cells itself. So it's the most sensitive assay to find it, which we can use for serum, plasma, or any other test method as well. We got really good sensitivity and for all the targets in presence of higher and lower target other virus existence. The third panel I developed, that is for the hemorrhagic fever and hemorrhagic fever-like disease in West Africa during the Ebola outbreak, because there are so many cases were coming to. It was very important to identify the people who got the Zaire Ebola, that they should be isolated, and also identify who did not get the Ebola, then they should not be put with the containment zone with other Ebola patient, so they may get infected. So we made these 2 panels, and that identify all those viruses that can present very similar clinical symptoms early on before the like Ebola and Lassa. They can lead to hemorrhagic fever, yellow fever is circulating, dengue can and dengue and plasmodium can give early signs of like the febrile illness, salmotype and Shigella. They can give the fever as well as gastrointestinal changes as well. And we have the housekeeping gene here as well. So these are 2 panels which we worked up on that to identify all of these agents. And all of these agents work like with very high sensitivity. And this was done with the Google Foundation for the West Africa and with NIH. So these are all methods I mentioned. You all know about the qPCR now. We know about other acute diagnostic assay also. They are antigen-based tests, the lateral flow assays, they are RT-PCR. I will not go into those details, but I'm happy to talk about it if anyone is interested about that. The next one is high throughput sequencing. How did we make these qPCR assay, or either us or somebody else throughout the scientific community, they were able to provide us genomic sequence of these viruses or bacteria. Once we have those targets, then it's convenient to make a new diagnostic qPCR method which can use worldwide. But how do we identify those viruses? We discover the new disease, and then we need to know which bug is circulating into that community that is causing the new disease, and then after that, we need to identify the total complete genomic sequence. So, so there are two parts of that. One is discovery, identifying the pathogenic agent, and then second, characterizing and putting the full genome out there so we can work up on the research in the future on that genome. So let's say it's a simple scenario which I always think about. This is a clinical sample. We have like virus and bacteria, fungus, um, coming out from the different part of the world. It's part of the environment. They are be the commensal bacteria. They are part of the water we drink. The bacteria are on the salad we eat, and all of those things are there. And we have maybe like some of you have seen the recent diarrhea and the recent enteric outbreaks happening throughout the world because of some contamination of the food as well. I go out, I eat my lunch somewhere, and then I meet with multiple people. Among those all virus and bacteria, this is one is pathogenic, which can make me sick tomorrow morning. And among these, like the cloud of so many virus and bacteria, identifying this one, either I do PCR for everything. It's cumbersome, it's very difficult to do it, and it's impossible if we do not know about this pathogen as well with our clinical symptoms. And this scenario can happen in the human clinical sample as well. It can happen in the wildlife also when we do the surveillance. When we look for the bats and rats and rodents and other animals, how much virus and bacteria they're carrying. They can happen in the marine life also. The fish and other crustaceans, what pathogen they are carrying or what other microbial agent they are carrying around. So this could be infectious bug. How do we can identify? So there is one approach. We take all the nucleic material and do the unbiased sequencing approach. We will generate all the data what we do not need, and then we have to identify some sequences that are related to this infectious bug. And it's like literally searching for needle in the haystack. Or what we can do, put an applied approach where we have these like all the high throughput sequencing, but where we can create a magnet kind of thing that is pulling out all the viral reads only, or pulling out all the bacterial reads only what we need. And then after the magnet is stuck to all the viral bacterial reads, it's easy to identify, and it will be putting less effort on the high throughput sequencing library preparation as well as downstream data processing only. So the two technologies which I will talk about, we have developed in the last 5-6 years and very widely used. Vircapsic-Vert and Backcapsic. Vircapsic-Vert stands for virome capture sequencing for vertebrate viruses. That means it covers all vertebrate viruses what we could think of or presented already, and all the future vertebrate viruses which could be emerging or closely related with the pre-existing viruses around the world, which have been identified, of course. And second, bacterial capture sequencing, which can identify all pathogenic bacteria. Especially, it's very important because bacterial identification takes 3 to 4 days in the clinical setting, and it's a very long time. And during that process, the wide spectrum antibiotics has been given to the patient that can lead to the antimicrobial resistant gene. So the culture process is long, it take a while. What if we switch it to the sequencing process, which can happen 24 hours? Not only it can provide us like the specific bacterial sequence, which is causing bacteremia, probably it can also tell us which kind of antimicrobial resistant gene is carrying. Based on that, we can decide is wild or is resistant, and the antibiotic can be chosen accordingly rather than throwing the broader spectrum antibiotic package onto them. reiterating what I said, is the virus capsid word. It can identify all the viruses throughout the world. So we do not need to do a qPCR or real-time PCR culture or staining for so many viruses at the same time. It can be applied on all kind of samples: plasma, serum, tissue, urine, semen, um, any, or also environmental sample, wastewater, or any other thing. So how did we make this? So what we did for VirCapsid word, we made like 2 million probes, which stands around 100 to 120 base pair. Those probes are distributed throughout the virome of all vertebrate viruses, everything. And same thing we have done for the bacterial cap sequences. We have identified those island motif. Those are very conserved because bacteria can be very homologous among them because they share the genetic sequences. So we look for the only specific regions. Those are distinguished for each and every bacterial strain. We made the probes for that, and we also made the probes for those antimicroresistant gene for that. So there are 4.2 million probes for the back capsid. Um, this is like a scenario showing why do we need to know AMR gene. Most important, not only just culture cannot tell us if which AMR gene is carrying the bacteria. So that is very important because antibody resistance is like threatening emerging pandemic, which we know of, but we do not consider as a very active pandemic as well. And we are increasing antimicro resistant bacteria all the time. So how does it happen? So what we have done, we took the exome target or like whole proteome targeting like from the viruses and especially from bacteria, and we designed the probes like that. So the probes cover the whole virome for each and individual virus, and then after we add a biotinylated probe on these probes, biotinylated junction on this probe, and then after we use them for capture. Why it is important? If we have to take any clinical sample from human, let's say we take a fecal sample or tissue sample, what it will be consisting if we do for next generation sequencing or Illumina sequencing or high throughput sequencing, which is commonly said, majority of these sequences are coming from the host genomic read as well. A lot of bacterial sequences. Bacterial sequences can happen on the top of a skin surface. There are other commensal bacteria that live in our gut and oral microbiota as well. There are bacterial sequencing in the reagent as well. Nothing is totally sterile. Then there are fungal sequences that can come from the environment, on the skin, on the hair, and the very small other environmental sequences which we don't know about. They may come from the food we eat, the bacteriophages that live in our gut, and then very small fraction of the whole sequences what we generate from a high throughput sequencing could be the viral reads, and after that very small percent the viral sequences which we want to target. So among this the whole crowd, the viral sequences are very smaller subset of that. Let's say we generated 10 million reads per sample. Out of 10 million reads, 9.5 million reads could be junk for our purpose, which we don't need it, but the high throughput sequencing unbiased approach is going to provide those reads. If the viral titer is not too high and or the samples like cerebrospinal fluid or plasma, we may totally miss out these viral reads because mostly is populated by host and other environmental sequences. So what we have done, we created the capture probes for VirCapsid word, which can increase up to 10,000 fold increase in the viral reads by the virus capture sequencing word method, and which can provide us 1,000, 10,000. This is like from the tissue sample, and we have spiked the viruses. And this is comparison with next generation immunosequencing on the same platform. With using the VirCapsid, we can see 1,000 to 10,000 fold increase in the very complex background. In the serum and plasma, sometimes we see 100,000 fold increase because we do the enrichment, we remove these junk sequences. So how the whole process works, this is small cartoon we made. We take the plasma sample, then we extract total nucleic acid. We make first and second strand cDNA. We like always doing DNA and RNA both because RNA can tell us like about RNA viruses. And second thing, it can also tell if there are mRNA transcript for the DNA viruses as well, which can lead to information is if it is actively replicating or is causing the infection at this point. Uh, Up to this point, it's very standard process for high throughput sequencing. We make double-stranded DNA, we do cDNA fragmentation, adapter ligation, and we do the qPCR on to know how much each library we have. But then after what we do, the 2 million probes we have made, we hybridize our library with those probes. And those probes are bioattenuated. After a couple of hours, we pull out this whole junction molecule that is attached with the bioattenuated probes for viruses and the targets if they are present into that. So what it does is pull out all the viral reads from the system, from the sample itself, and leaving behind host, bacteria, fungal, other environmental reads back into the wash. And what it does, so then we reduce the host background and other background, we enrich our virus genomic sequences, and we do the next second PCR, and that leads us to and then we do the sequencing. So this is extra process for the capture sequencing, which leads us to phenomenal result in last many years we have been using. Um, Workflow, you can look into that. I will not go into details. We try to generate 10 million reads per sample, and VirCapSeq can provide equivalent result what we could get it from the unbiased high throughput sequencing, 100 million reads. So what it does, it can be more economic, more financially accepted into the wildlife surveillance and wastewater surveillance, and it can provide multiplex data. We will know about all the viruses present into the sample. Because of an enrichment-based method, we can recover the full genome that helps in characterization of the virus, and we can also know what mutation are occurring and are they gonna immune escape for the current vaccination or current therapeutic program. Um, since it's a DNA hybridization method, it can tolerate the mismatches. So if the virus is relatively new from any existing virus, only 20% is matching, 30% is matching, it will always have some conserved region, and that will hybridize with the virus probes for that region itself. We might not recover the larger chunk of genome, but we may recover some RDRP polymerase helicase or some other conserved domain of any virus. Um, Once we identify that, then we can take that sample itself and we can recover sequencing, or we can try to culture, and we can do the unbiased sequencing. We can do other tools to recover the whole genome. This is the rotavirus genome from a bat, which is which has highly mismatched, 47 to 93% different fragment. And by using VirCapSeq, we could recover most of the genome using the capture sequencing. So it's very feasible to use this tool for the pathogen discovery as well. So what the recursive word does, this we published in many years ago. Increase sequencing depth, increase sensitivity, replacement of other target enrichment strategies, selective and vertebrate viral targets only. We do not want to know the plant viruses. We do not want to know the insect viruses. We would like to know the virus which can jump around, cause the zoonotic disease. That's why we are focusing vertebrate viruses only. And economic high throughput application through multiplexing. I will tell you about one recent work we have done using the VirCapSeq. There was like patient who were showing some skin lesions, and they were all in solid organ transplant patient, skin lesions, and slowly they have the respiratory issues, and their whole health has been declining with the time. There was suspect of polyomaviruses. All the tools has been taken, BioRad and other biofire assays, and the IFA and antigen tests. They could not discover any microbial agent. Then they reached out to us, and we put the sample for VirCapSeq analysis. We identify in 3 patients. These are skinner macular rashes, and 2 patients were actively enrolled during the study. And the first one patient, which was already dead, like a year ago, we also included that in the study. We identify human polyomavirus 9 into the skin and the plasma of these patients. Then we did the further histopathology. We could see the virus into the skin cells and other cells as well. Uh, since like they already been going through the typical clinical scenario, uh, it took us like 1 year. It took us 1 year to get us the samples itself, and they had to reach out after 1 year. Unfortunately, all three patients died before we could identify the virus itself. We could identify human polymer 9 was pathogenic into these patients. Generally, this is a common virus. It can live on the skin and shed, but in immunocompromised patient, it could be very pathogenic in some patient. All three patients died because of pulmonary failure. One patient we could do the autopsy also, and we could detect the virus into the lung tissue as well by the high throughput sequencing, and also we had confirmed with the in situ hybridization. So what it did, we could get to know this is the virus which is causing very specific type of clinical illness into these patients. We made a qPCR assay on that basis. Soon after, 5 other patients have been enrolled in Colombia with me and my colleague in the dermatology. Um, since we know about these virus now, so we can detect them, and that we can also look for their viral load regularly. And based on that, they started cidofovir treatment onto that. The first 3 patients who died within 12 to 14 months of the skin eruption. Other 5 patients, they are surviving up to 2 and 2.5 years. Whenever their viral load increase, their cidofovir treatment increases as well, and the viral load come down. So for their living with the virus infection, the persistent viral infection is very difficult to clear up, but still this whole process could save other 5 patients which could go totally undiagnosed and dead because of this viral infection in the solid organ transplant patients. Only problem, cidofovir can cause renal nephropathy, so it's not a sustainable plan. So based on that idea, we are working with a different group. We are looking for some CAR T therapy, or we are looking for some antiviral therapeutic antibodies. SARS-CoV-2, it's difficult to identify the full genome of SARS-CoV-2, which is very important because we want to know what mutations are occurring, which is the new variant, how it's going to play with the current and existing vaccine and other monoclonal antibodies. Using the Virchow-Sigwart approach, you can look at the upper panel. When we do unbiased sequencing, up to 25% in one sample, this is the total viral reads, but majority of reads were in compared to were from the an unbiased system with the gray bar, and the green bar, the high throughput sequencing with the Virchow-Sig. You can see the increase up to 100-fold. And the next panel, you can see the vertebrate genomic sequence we get in swab sample. Those have been decreased from 80% to up to 15-20%. So what it does, it provides us very good coverage, it provides enrichment, and we could recover many times the complete genome sequence also, even the CT value was 34. Recently, if we know about, like, there was surveillance of SARS-CoV-2 in Hunan seafood market. They also use our Virchow-Sigwart platform, and recently adeno-associated virus in children with non-A hepatitis also. They also use our Virchow-Sigwart platform. Search it on the PubMed, you will find more than 40-45 publications where we have the Virchow-Sigwart. There was a patient who came back from Vietnam and with suspected meningitis. We have a grant from the with NINDS and multiple other centers. We look at this patient. We couldn't identify anything cerebrospinal fluid. It's for like 4 or 5 years. This person living with the persistent headache and some neurological symptoms. We applied that CSF sample on the Virchow-Sigwart, and we could recover dengue 1 isolate. When we looked at through where this genome come from, this genome was come from the Vietnam. And when we look at the patient history, he was deputed at Vietnam. He was working with the Namru, and he was deputed to Vietnam, and he told his clinician he got some dengue viral infection 5 years ago when he was working in the Vietnam. So probably that time only the virus made it to the brain and living persistently there, and then this really literally helped this patient as well. I was working in India for acute encephalitis syndrome. that was causing more than 25,000 death in last 10 years. Every year, several hundred patients, they get infections, generally children with acute encephalitis, unexplained. We took the cerebrospinal fluid from the multiple ears. We processed them with Vircapsic, Vert, and Baccapsic. And in the undiagnosed sample, which have been gone through the whole panel from CDC, 50-60% sample, we could identify at least one pathogen. The major pathogen which were undiagnosed, but they were Japanese encephalitis, Orange Succiumasi, Typhus, rickettsial, and enteroviral infection, um, which could be, these patients could be easily saved if we were using this modern method of identification, not only the qPCR methods. Recently, we have obtained the CLEP certification for Birkap-Sigwart in the serum and plasma to use for clinical diagnostics, and we are working to get the CLEP approval for other tissues as well. And this test can be used with the Laboratory of Personal Genomic Medicine and with CII's effort, and this was one of the world-changing idea in 2015. I will not spend a lot of time on Backup-Seq. It's a very similar platform where we are targeting only island motifs only, and then we can identify the complete genomes. You can see orange versus blue. Without Backup-Seq and with Backup-Seq, the enrichment of reads happen, and we can also identify the antimicrobial resistant gene based on the number of reads increase. We can identify which AMR gene is carrying with this bacteria, which can help in the therapeutic. Recently, with collaboration with the encephalitis group, neurology group in Colombia, and pediatric group in Colombia, we published 2 papers where we have applied Backup-Seq for identification of bacteremia in cancer children, and also in the definitive neuroinfectious disease, we have applied Backup-Seq and Vert both, Backup-Seq both. Based on that, like, we have been making the tools for diagnostic for a long time for characterization and sequencing, mostly samples we receive. But in many years, we were also going to the field on the invitation, like when I mentioned acute encephalitis syndrome. We were invited to India to investigate that unknown, unexplained encephalitis. Similarly, many years ago, 2013, we went to Saudi Arabia when the primary MERS case arrived. We set up the lab over there, bat capturing, camel sampling, and other wildlife sampling to identify the source of the MERS itself. We made the mobile laboratory, went down there, do the qPCR and everything for high throughput sequencing. We got samples over here, we sequenced them. During the qPCR only, we identify how the virus traveled to the camels. We identify the one incident hypothesis Preparedness bat. So probably the bat infected human, camels and camel infected human, and camel human and human to human infection were happening for a long time. So we could find the reservoir of the original MERS coronavirus. Then once we identify that, we look for the sero survey and the molecular survey in the camels, and we can find up to 95% adult and 55% juvenile camels. They were infected with the MERS coronavirus already, and if it increases from juvenile to adult phase, that tells us like earlier they are not infected within living in the camel fair or camel markets. The virus can spill to camels and spread it very widely, and then it can jump to human when they're living with the comorbidity or older people. Molecular investigation told us up to 35% juvenile and 15% adult. So you can see the number are switching. That when juvenile get infected and they produce the antibodies, and that lead to the protection in the adult, which is common. So we did not find many adult also infected with the carrying virus. That when they build the immunity. When we looked at some blood bank for camels they have, because camels are very broadly used, we could identify at least 15 years before the MERS emerged in the human sample. In a human case itself, we could find the seropositivity in the camel. So the virus was circulating camels. One day it found the right time and right mutation, they switched to the human, and they adapted to the human race, humans as well. We published several papers on that, identifying the source of the dromedary camels for the infection. We also looked at how the virus mutated and adapted to the human population and is circulating in the human itself. And we also did viral metagenomic survey to identify if there are some other viruses circulating in Saudi Arabia. So this kind of effort we have done several, but recently we made this program from 2018-19 onwards, Global Alliance for Preventing Pandemics. This is a global program. What we do, we help the countries which are underprivileged, or they lack resources, or they do not have infrastructure. We bring them over here. We train them on these tools for high throughput sequencing, analysis, pathogen discovery. We help them working on their own samples, and we help them analyzing the data, storage the data, and as well as publishing that work, so they are prepared for the next outbreak. And then after, we help them to set up the laboratory on site. So we have a lot of effort on the CIN gap. So you can see the right panel. We work on automatic nucleic acid sequence serological analysis. We are making the simpler platform for them also. They can analyze. We are trying to create a network when in the future, in a couple of years, the country we have trained, they are able to train the other country, or they can make a center for that. It could be part of Africa, it could be Asia, South America, anywhere in the world. So mostly our goals are biosurveillance to identify the new emerging pathogens, discovering by training, sequencing, serology, and data analysis. And then after, they can work on the on-site education. We can help them remotely. They can continue working on the ground in their own country, in their own centers, and then build the whole system itself. So we want to replicate multiple efforts throughout the world. So in the next pandemic, like I said, we can stop it outbreak level only. We can contain it as soon as possible before it emerges. This is our presence for the gap already. South America, part of like India, Bangladesh, China, Indonesia, Western African country, Mali, Uganda. All of these teams have happened to be with us. We train them, we work with them, and they are processing their data. And we will be seeing more publication from this collaborative effort very soon. These are two interesting studies when we are building the gap. I work with the two countries. One is Liberia, one is Mongolia. Liberia was like case surge after the sudden, mostly they did not have many cases. All of them, they have sudden case surge. And then we investigated these samples. The person from Liberia came to us with the samples. We sequenced it. Within 2 weeks, we could tell them this was a Delta virus emerging. That's why they were having the higher surges of the cases. Interesting was Mongolia. Mongolia was 65-70% vaccinated. It was the first country who vaccinated 65% of their people along with the America also. They also slowed down the restriction policies over there, and all of them, they also have seen sudden breakthrough infection in the vaccinated individual, more hospital cases, more mortality, which they could not understand. Earlier, they were suspecting the Delta virus inclusion in Mongolia, and which the vaccine was not effective. We did sequencing, we did the serology with them, and we identified this. Still have the alpha virus, alpha variant research, and the breakthrough infection happening mostly with alpha variant only. The vaccine is they were using to immunize 65-75% population. Those vaccines were not as effective with the current back then current variants. So they were not providing very adequate protection against the breakthrough infection, and that's why they are seeing more hospitalized cases. And that lead to the public health policy working with them, and then they gave them all of their individuals 1 dose of Pfizer booster only on the top of attenuated vaccine, and they could go through from this crisis as well. So far, we have talked about mostly about molecular approach, about how we can identify viruses, how can we identify bacteria, how can we make the tools that can help in the identification, discovery, and protect and surveillance of these viruses and bacteria in the humans and wildlife and animals itself. But what if, like we talked about it, what if like molecular genetic material is not present or is subacute infection? We want to say how viruses are spilling over from one population to another, from one species to another. What are the chances of like future pandemic if there is protection, or the bat and rodents, carry hundreds of thousands of viruses. Not all the viruses are infectious to human, um, but we should be, and it's vice versa also. There is a reverse zoonosis as well. So what we could do, rather than looking for the nucleic material itself or genetic material, DNA and RNA, we can look for the indirect approach. Like, let's say there's a specific bat virus or like circulating rodent virus, which could look like very pathogenic. We can look for the antibodies against that virus in the community and people and where it has been identified. So for that, we have been working with this high throughput serology approach, peptide microarray for linear discovery platform. uh, how does it work? So we have like very small glass slide, that the pathology slide we see, and that slide can carry up to 6 million peptides of 12 amino acid. So all the targets which we want to cover, we take the whole proteome, not the genome, whole proteome of those viruses or family or bacteria. We make this sliding window peptides, um, and then after we synthesize this peptide and print on the small glass slide. As we know, antigen and antibody interaction site is epitope, generally 6 to 7 amino acid, and our peptides are 12 amino acid long. So any it works on the linear epitopes, but anyhow the antibody can bind to this specific epitope, and all the epitope has been distributed throughout the chip, and they are on the different spots. So like uh, +1 and -1, they are not together. They are printed on different spots of the array itself. Then we add the plasma sample, serum sample onto that, overnight incubate. Then we use secondary antibody and take the fluorescence picture on the microarray scanner. And we have the virtual design that is given by manufacturer, that tells us the coordinate of each and every peptide. And then virtual and the image file, we overlap. And all the fluorescence we convert to some arbitrary number. Higher the arbitrary number, higher the chances of antibody bind to the specific antigen is part. So what it provide us very specific binding site. So we do not need to use the whole viral ELISA. We do not need to use the recombinant ELISA. We can identify the specific immune response among multiple very similar viral species, like from Zika versus dengue, SARS-CoV-2 versus SARS-CoV-1, enterovirus A versus enterovirus B. So it is very effective because if we take the whole protein, these viruses can cross-react with each other with antibodies. But when we take those only epitope binding site, we can identify distinguished, very differential epitope among these viruses. Using that, there was a uh, acute flaccid myelitis was happening from 2015-16 in America. More than 700 cases has been reported. Um, Majority of time is very polio-like illness in children, but never like only 1 or 2 cases enterovirus 71 or enterovirus D60 have been identified in cerebrospinal fluid. So generally, infection start with the respiratory route, and it can lead to the brain. And when the neurological symptoms start coming, virus disappear from the system itself and just causing the hit and run mechanism, aftermath damage only. So only way of looking for the antibody response. So we created a peptide macroarray that covers all enteroviruses and the common pathogens. When we detected cerebrospinal fluid of those patients, acute flaccid myelitis patient, we did not identify any molecular signature Viber-capsid, but we did identify higher amount of antibody against enteroviruses, especially enterovirus D68. And then we could look into that AFM, CSF, and sera sample, that provide us information. The enterovirus D68 is the major pathogen that is causing acute flaccid myelitis in the children in the United States and worldwide, and that provides a target for looking for the future cases. We can take this peptide only, we can generate a peptide ELISA and detect it very quickly. We do not need to go through this whole discovery platform. And this also provides us target to make a lateral flow diagnostic assay as well as therapeutic antibodies. We also did some SARS-CoV-2 work. It's very difficult to identify. The coronavirus also present like greater homology between different proteins. And as we know, a spike can be positive after immunization, but the immune response changes with the time. So we put the whole human coronavirus proteome on this coronavirus peptide arrays, identify several epitopes which are related to severity of the disease as well and recent infection. Those were throughout the proteome, not only spike and nucleocapsid. Those covers from the different part of proteome of non-structural protein as well. Once we identify these proteins, the peptide signature, we can detect them. There are some spikes, some are playing the neutralizing role, some are not. And once we have identified these immunogenic peptide, we can convert it to some simpler fat form. Recently, we wanted to work with collaborative project with NIH and Liberia and USDA. There we wanted to look for the current filovirus vaccine trial, Ebola virus vaccine trial. We wanted to distinguish how we can distinguish people uh, who had been vaccinated one dose or two doses and have been immunized and or infected also in the past, which was asymptomatic. Using that, we have taken some macaque experimental samples and we tested them the filovirus peptide array. We found these distinguished immune responses, and we tracked those epitope also highly immunogenic. That tells us the immune response is based on the dose of immune dose, the vaccine dose is given, as well as the time. As the time progress, we can see the higher immune response in the red, and we have tracked down these all the peptides as well. We are further working on the diagnostic approach as well as we are looking for the characterization of post-immune and post-infection responses. Like I said, this is our discovery platform. Once we have this platform, we can take this peptide, like in working with Zika and dengue. Since we print the whole peptide, whole proteome of Zika, dengue, and all the viruses, generally for flavivirus target for serology is NS3, NS5, or envelope protein, but we could find a very distinguished immune response which was not cross-reacting with other flaviviruses or Zika and NS2B. We take this peptide and we made the peptide ELISA that provide really good sensitivity up to 94%, and that can help us to for epidemiological study as well as recent Zika infection as well very rapidly by the peptide ELISA itself. And SARS-CoV-2, I said, what we have like identified like 63 peptide, we convert to Luminex assay, and Luminex assay provide us very good serodiagnostic in compare higher than native virus, pseudovirus, ELISA, and spike trimer ELISA. We could find up to 92% sensitivity and 100% specificity. We can see different peptide enriches at different time point after post-infection. We we are expanding this work. We are trying to find if with the peptide reactivity, we can predict how long ago the person had the actual infection, because it's very difficult to identify once we identify antibodies, your antibody decline with the time. And that can help if they need a vaccine booster dose or not. This is like same data. You can see the specificity increases with the time and the decreases with the time to 16 weeks. We have used this peptide arrays for different approach. I did not mention the most important work that we have done with tick-borne disease, the 7 tick-borne disease targets, including Lyme disease, Borrelia, Babesia, Powassan virus, and we are working on lateral flow assay for providing actual serodiagnosis for tick-borne diseases, and that we are working with our partner who make the lateral flow assay. We have already identified these peptides. Okay, and then we have several other serological assays which we're working on to identify the diseases. Our like next project is we want to make a zoonotic array. That's a next generation serodiagnostic that will be multiplex, high throughput serology that will cover all viruses which can be emerging or called the chronic infection. And these viruses are very high in number. It's difficult to accommodate in 6 million peptide, and printing multiple peptide array is difficult also because they are very expensive. So we are going to short peptides phase display approach. Phase display is based on the bacteriophage, which is bacteriophage is a virus of the bacteria. So simply I will put it through there are two different M13 is a thread and the T7 phase, which are two phases can be used. We are using T7 phase. People who don't know about it, since we have already peptide microarray data, so we can compare what is the best length of peptide. We are taking 16 amino acid peptide because it can provide a very distinguished immune response between closely related pathogen as well. And we are incorporating this peptide to the bacteriophage genome, making a recombinant of that. And then after we reinsert this genome to bacteriophage capsid, so itself, and then we grow them. Once these bacteriophages start replicating, they will express their protein as well as they will express our inserted protein. In the T7 phase, our insert point, it comes to one of the leg of the bacteriophages, and it will express our protein of choice, and the 16 amino acid will be expressed. And if we identify any antibody to the sample, antibody will bind with this antigen which is expressed on the bacteriophage tentacle. And after that, we can pull out this whole combination antibody plus bacteriophage, extract nucleic acid. The nucleic acid expressed on the tentacle itself will be the same sequence which was inserted in the beginning. And once we sequence that insert, we know what is the epitope or what is antibody binding site for these bacteria. Okay, so here I think I will take a break. What are our broader goals? We want to implement this technology as much as possible. We want to help people. We want to create a network onto that. We want to contribute to sustainable global infrastructure for pathogen discovery, microbial and serological methods. We want to help them identify the disease earlier they should or they can, and we want to create a sharing-based system where we can share the information, where we can share the genomes, where we can share the other information, other knowledge about the viruses and bacteria, so we know about any emerging pathogen in advance, and we are well prepared for the future when the next emergence happen. Okay, and here I will take my break, and thank you very much. I'm happy to take any questions, and thank you everyone again.
All right. Well, thank you so much, Nischay, for that informative presentation. Unfortunately, we are out of time for today and will not be able to answer our audience questions during this live session. But before we go, I'd like to say thank you again, Nischay Mishra, for your time today and your important research, and to the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until September 7th, 2025, and Labyrinths will alert you via email when it's available for replay. So we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Thank you.